Have you ever stopped to think about how your body with its trillions of cells, we're talking brain cells, muscle cells, skin cells, all runs on a single instruction manual? It's kind of wild, but every single one of them started with the exact same 3 billion letter DNA code.
So, how does the cell know what it's supposed to be? That's the big question, right? And that's exactly what we're going to break down by looking at the central dogma of biology. My promise to you is to make this so simple, you'll walk into your next exam feeling totally confident.
So here's the game plan. We'll cover how that master code, the DNA, gets copied into a working message, which is RNA. Then, we'll see how that message gets read to build an actual protein. After that, we'll look at how the cell controls this whole factory. And stick around, because I've got a super simple exam tip at the end that's going to lock in a key concept for you. Let's do this.
First things first, let's talk about the master blueprint itself, DNA. The absolute best way to think about your DNA is to imagine it's a massive, priceless cookbook. It's kept super safe inside a library, which is the cell's nucleus, and this cookbook, it's got every single recipe or gene your body could ever possibly need.
So what's the stuff actually look like? Well, you've all seen that famous double helix shape. The outside railings are just a sugar-phosphate backbone, but the real magic, that happens on the inside, on the rungs of the ladder. That's where we find the 4 chemical bases: adenine, guanine, cytosine, and thymine. Let's just call them A, G, C, and T.
Now here's the key, and it's shockingly simple. A always, and I mean always, pairs up with T, and G always pairs up with C. That's it. That's the rule. These pairs are held together by these weak little hydrogen bonds, which is actually super important because it means the 2 strands can be unzipped really easily when it's time to read the code.
Okay, so if DNA is our priceless cookbook locked in the library, how do we actually use it? I mean, you're not going to take that one-of-a-kind book into a messy kitchen, right? No way! You'd make a photocopy of just the one recipe you need. That's exactly what transcription is. It's making a temporary, disposable RNA copy of a single gene.
And the little machine that does this photocopying, it's a superstar enzyme called RNA polymerase. It's like a molecular copy machine, and it knows exactly where to start copying because it looks for a specific start signal on the DNA, a little landing strip called a promoter.
So the process is pretty straightforward. RNA polymerase lands on that promoter, it unwinds a tiny section of the DNA helix to read the code, and then it just zips along one of the strands, building a matching RNA copy as it goes. Once it hits a stop sign, it just pops off, and that brand new RNA strand is released.
So what's different about this RNA copy? Well, there are 2 key things you absolutely have to know for your exams. First, it uses a slightly different sugar called ribose. And second, and this is the big one, it doesn't use the base thymine, or T. Instead, it swaps in another base called uracil, or U. So anytime the original DNA had an A, the new RNA copy will have a U. But hold on, there's one more little editing step. The very first copy the cell makes is kind of like a rough draft. It has a bunch of extra junk in it, stuff that doesn't code for anything. These useless bits are called introns. So the cell does this cool editing job called splicing, where it literally cuts out all the useless introns and stitches the important parts, the exons, back together. What's left is the final, clean recipe, the mature messenger RNA, or mRNA. All right, our perfect recipe copy is finally ready. It leaves the safety of the nucleus and heads out into the main factory floor of the cell, the cytoplasm. And what's its destination? A ribosome. And you can think of a ribosome as the cell's protein-building factory.
Now, the ribosome is smart. It doesn't read the mRNA one letter at a time. No, it reads it in 3-letter words called codons. Each one of these codons, like CCG or GAA, is the code for a very specific ingredient, one of the 20 amino acids that are the building blocks for every protein. To get this job done, we need a whole team. First, you've got the mRNA. That's our coded message. Then you have the ribosome, which is our factory. And finally, you have transfer RNA, or tRNA. These guys are the delivery trucks. Each tRNA is specialized to carry just one type of amino acid, and it has a little 3-letter key, an anticodon, that lets it match up perfectly with a codon on the mRNA.
And the whole thing works just like a perfect assembly line. The ribosome chugs along the mRNA, reading one codon at a time. The right tRNA delivery truck docks on, drops off its amino acid, and then the ribosome links that amino acid to the growing protein chain. This just keeps happening over and over until the ribosome hits a stop codon, which says, hey, we're done here, the protein is finished.
So, we have this incredible protein-making factory. But there's a problem. A cell can't just be making every single protein all the time. Think about it. A skin cell has no business making hemoglobin, and a brain cell doesn't need muscle proteins. It would be a huge waste of energy. So cells have to be able to regulate the factory.
And they have tons of control points. The biggest and most common one is just controlling transcription in the first place, basically deciding whether to even make the photocopy. But they can also control how that RNA gets edited, whether an mRNA that's already made actually gets translated, and they can even turn finished proteins on or off.
At that first level, transcription, think of it like this. The promoter is the main on-off switch. Then you have other bits of DNA called enhancers that can act like a turbo booster, really speeding things up. And the cell can literally lock the cookbook by wrapping the DNA super tightly around these proteins called histones, making it physically impossible for the RNA polymerase to even get to the gene. And here's a really cool, more subtle layer of control. These tiny little molecules called microRNAs. If transcription is the on-off switch, think of these guys as the volume knob. They can hunt down a specific RNA molecule that's already floating around and either block it from being read or just mark it for destruction, basically turning down the volume of how much protein gets made.
And there's also this really simple, elegant system for the finished products. In a lot of cases, once a cell has made enough of something, that final product will actually go back and shut off the very first enzyme in its own production line. It's called feedback inhibition, a perfect auto shutoff system so the cell doesn't waste energy making something it already has plenty of. All right, here it is, the exam tip I promised you. This is for remembering that super fundamental DNA base pairing rule. Just remember this phrase, A at the Grand Canyon. A from at pairs with T from the, and G from Grand pairs with C from Canyon. AT, GC. Easy.
And what about for RNA? It's even simpler. Just remember this, thymine takes a vacation and uracil, or U, takes its place. So in RNA, adenine pairs with uracil. That's all you need to know.
Okay, let's put it all together one more time. Here is the entire central dogma in 20 seconds. Ready? 1. Your DNA is the master blueprint locked away in the nucleus. 2. Transcription happens, making an mRNA working copy of a gene. 3. Translation happens, where a ribosome reads that copy to build a protein. And 4. The entire process is tightly regulated at every single step.
You know, this system is unbelievably precise. It works perfectly millions of times a day inside your body right now. But that leads us to one final, really important question. What happens when this finely tuned system makes a mistake? Well, that's where things like mutations come from, and that's when things can go very, very wrong.
If this helped you finally get a handle on gene expression, do me a favor and take 1 second to subscribe for more of these high-yield physiology breakdowns.